So great to have you here for our Science at Cal lecture on the origin of life with Dr. Charles Marshall. My name is Dee Rossiter. I'm the Executive Director of Science at Cal. I will explain what that is in just a second. We also have Roslyn here from Science at Cal. Say hi, Roslyn.
Hi, everyone.
Roslyn, where are people tuning in from? Where's the farthest place?
Lots of places.
Okay.
We have folks joining us from Malaysia all the way to Australia, Brazil. One of my favorites is Interstate 80 in California. Maybe people are joining us in their car.
That's great. We had somebody from Australia last time, although this time it's less shocking, right? Knowing about our speaker. All right. Thanks so much, everyone, for being here. We're so excited to have you. Let's see, we're already up to 140 participants, so that's wonderful. There's a big crowd here today to meet Charles and listen to him and his science. So let's get started. Before we begin, I do want to do a land acknowledgement. We recognize that Berkeley sits on the Huchun territory, the ancestral and unceded land of the Ohlone people, the successors of the historic and sovereign Verona Band of Alameda County. This land was and continues to be of great importance to the Ohlone tribe and other familiar descendants of the Verona Band. Every member of the Berkeley community has and continues to benefit from the use and the occupation of this land since the institution's founding in 1868. By offering this land acknowledgement, the Berkeley community not only recognizes the history of the land on which we stand, but also recognizes that the Ohlone people are alive and flourishing members of the Berkeley and broader Bay Area communities today. So thank you for allowing me to take the time to do that. Science at Cal, as it states bold and bright here, we celebrate science. Science at Cal brings the wonders of and excitement of UC Berkeley STEM, that's science, technology, engineering, and math, research to the community. All of our events programs are free and geared towards public audiences. So we are so happy to have you here. You may be part of the Science at Cal family and join us often, and you may be new. Either way, welcome. We are happy to have you. We do programs, science cafes like this one. When traditionally we're live, we would meet all of you in person and we would have you in our home at UC Berkeley or across the East Bay in different venues where we get people out to talk to scientists one-on-one, maybe at festivals, Cal Day, and more. We have campus and community partners across the Bay Area. Sorry if you hear my background noise. And we have a few upcoming lectures. Our next Science at Cal lecture is on the Oakland Eco Block. So we hope for you to join us there in April. So save that date. You'll be learning more. We'll be sending out registration about that soon. We also have a Grounds for Science, which is our lecture series that features 2 graduate students. And this one is going to be on public health, representation and research, and also the good side of viruses. So that's next week. If you haven't signed up, please do. And we have our next Midday Science Cafe, which is our event with Berkeley Lab. So please join us for that as well. I want to thank you, all of our donors, all of the people who support Science at Cal, either monetarily or, our faculty who supports us, everyone on our advisory council, also the Lawrence Hall of Science, thanks as well. And before I hand things over to Charles, I'm going to stop sharing my screen, but I do want to say that, and let him share, I do want to say, like I said, we're going to keep all of our videos on mute. If you want to clear up some bandwidth, you can also turn your video screens off. I will be doing that. Please continue to, comment in the chat, ask questions for Charles in the chat, but we are going to save all of the questions for the end of his presentation, just so we have a time to have that conversation. And Rosalind and I will be recording all of your questions, so we make sure that we get to them as they are asked. And then also, if you want to engage and ask Charles your questions directly, you can go ahead and if you see at the bottom of Zoom, there's a reaction button. You can actually click the reaction button and click raise hand to ask questions. But again, we're going to save our questions until the end of Charles's talk. And also this program will be recorded and we will provide it for you in a few days after we have time to wrap things up. So I'll go ahead and begin my introduction of Charles, and you can go ahead and share your screen whenever you like. So Charles was an undergraduate at the Australian National University. He did his graduate work at the University of Chicago and at NIH, National Institutes of Health, postdoc at Indiana University. He then spent 8 years in Earth and Space, sciences at UCLA, becoming tenured in 2 years. This was followed by 10 years in organismic, I knew I was going to mess that word up when I got to it, and evolutionary biology, and earth and planetary sciences, and at the Museum of Comparative Zoology at Harvard University. He has been at Cal now for 11 wonderful years. He has received many accolades, many awards, and he serves as the trustee and president of the California Academy of Sciences, on the board of the Berkeley Geochronology Center and on the board of reviewing editors for science. He is an unusual paleontologist with training in mathematics, the physical sciences, geology, and organismal and molecular biology. His broad research interests center on understanding the nature of evolutionary innovation and extinction, where his work often involves epistemology. No, I had it earlier, Charles. I had it earlier. Okay.
Epistemological.
Oh my gosh, epistemological. Oh, I did it with such ease earlier. Innovation. He enjoys teaching. I should have taken it out, including a previous course in dinosaurs and at Cal, a co-taught course on the origins from the Big Bang to the emergence of humans, which is what brings us here today. Sorry, I butchered that amazing and beautiful word, Charles, but I will let you take it away and probably say it so seamlessly. So thank you for being here and have fun.
Okay, thank you very much for the introduction and welcome everybody. Can you all hear me?
Yes.
Okay, excellent. So this is one of the strange things of COVID, of course, is the power of Zoom. So g'day to Australia and the rallies there, New Zealand. Salamat datang, Malaysia, hi to Singapore, the rest of the United States. It's good to sort of see everybody and sort of not. So what I want to do today is talk to you about the origin of life. And I want to explain, first of all, how it came to pass that I even wrote such a lecture. So a few years ago now, the Berkeley campus instituted a new undergraduate program for non-majors called the Big Ideas. And they had to be big ideas courses crossing departmental boundaries. And I was contacted by an astronomer, Elliot Quartet, to teach a course on origins from the Big Bang to the emergence of humans, which is what we did. However, you can't teach a course like that without considering the emergence, the origin of life. And I was completely dreading it because my sense was that we really had no idea how it happened yet. Some individual steps, yes, the overall arc of how life originated, no. And so my sense was that this hypothesis for the origin of life was about as good as any other hypothesis from the origin of life, plus or minus the few things that we think we already understood. My time at UCLA was spent in part at something called the Center for the Study of Evolution and the Origin of Life. And there I made good friends and co-taught a course with Cliff Brunk at UCLA. So I emailed Cliff and said, has anything happened in the discipline since I left UCLA? Cliff was apoplectic. Has anything happened? He said, has anything happened? You better believe it. And he sent me a deck of slides and I was stunned. It looks like we have an overall sense of how life originated. And what I wanted to know is, why didn't I know? It just seemed crazy. So what I wanted to do today is give you a sense of what that story looks like as much as one can in 40 minutes. So understanding the origin of life is a very hard problem, in part because organisms are very complex. And here's sort of a compound image of just showing the molecular complexity within the cell. And this is only part of it. Moreover, if you think of the way the cell operates, we have a highly complex integrated metabolism. And the next slide is a chart of your metabolism inside your body. So enormously complicated. So that makes it hard. The second thing is we only have data for one origin. If you're an astronomer and interested in the way stars operate, yes, we've got one close star, the sun, but we've got hundreds of millions of stars all around us that we can look at, but we have just one origin, no repeat experiments. Moreover, it happened a very long time ago, in the order of 4 billion years ago. Most of our record of the past comes from the geologic and fossil records, and those become increasingly poorer the further we go back in time. So we don't have much data. Moreover, the conditions on Earth were fundamentally different from now, and there are two that are particularly important. The first is there was no free oxygen, which is highly destructive of complex molecules. I don't think life today could originate on Earth because of the presence of oxygen. The second one is there was no life. Life is extraordinarily good at vacuuming up resources, but that didn't exist back then. So it means that some of our intuitions about the way the world worked don't work 4 billion years ago. Okay, one of the first ways we look at examining the origin of life is start with living species and work back in time to see what they have in common. And this turns out to be very fruitful. So there are about 24 groups of organisms that have become multicellular. The three big groups that we know are plants, fungi, and animals. And so what we can do is trace back their history to some sort of last common ancestor, and it will have looked something like this. A typical eukaryotic cell named after having a nucleus where the DNA is housed. Now, it turns out that if one then looks at truly invisible life, these things are in the order of 20th of a millimeter across, so just out of visual acuity. But if one looks at smaller things, one discovers that there's a whole range of other organisms that are invisible, and those are the archaebacteria and the bacteria, E. coli, salmonella, etc. And they are tiny. Your mitochondria turn out to be a bacterium, and there's one there indicated in red, and if I now eliminate the eukaryotic cell, that's its size. Way smaller, way simpler. A eukaryotic cell has about 1,000 times more DNA and is able to produce about 1,000 times more energy. So these prokaryotes, are rather simpler, but nonetheless enormously complicated. So working down the tree again, we can identify the last universal common ancestor of all currently living things, the red dot there. And so the inference then is based on the tree that the last common ancestor of everything was a single-celled prokaryote, like the one that I just took off the screen. However, modern life is still very complicated. And so it seems likely that the first organisms were much simpler. And it's even possible, hint, that they may have been fundamentally different from the last universal common ancestor, which is what I think we'll find by the time we reach the end of this. Finally, I'm going to offer you a tease. It turns out the archaebacteria, the archaes and the bacteria, the cell membranes and the cell walls are made of different materials. There are several other differences as well, and that's going to turn out to give us enormously important information for making inferences as to what LUCA was really like, but we're not there yet. Okay. Our attempts to understand the origin of life, I feel to some extent, has been misdirected by our knowledge of living organisms. Almost all life today depends ultimately on sunlight via photosynthesis. We now know that's no longer true or necessary. All life today is built around DNA and proteins, and all life today is made of cells. But we're beginning to shed the shackles of these assumptions, and that's partly what I want to examine in the material that follows. Okay, so to explain the origin of life, it would help to know a little bit about what life is. I'm trying to avoid definitions because they're very brittle. I might describe a lobster as a 10-legged crustacean. If I then rip a leg off, is it no longer a lobster? Nonetheless, when one thinks about life, one tends to think about organization, about growth, about information. The problem with those sort of definitions is you finish up with, oops, objects like this. Crystals grow, they're highly organized, they're not lifelike, partly because they don't reproduce in the standard way that we do. This is pictures of giant crystals in a subterranean cave in Mexico where the water's been drained out. So also, as one of my graduate students, Tanner, pointed out when we went through a rehearsal of this a couple of days ago, life is really a verb, not a noun. And so to explain the origin of life, we really need to know what living is. And so here are some components that I think are important when thinking about living. So what does it mean to be alive? So organisms are well-defined corporeal entities. We are material, we have insides and outsides. We are dynamic entities, like a vortex. We depend on energy flow in and out. We depend on material flow in and out. But unlike a vortex, our inside state is very different from the outside state. We are responsive, which usually means altering our internal state in response to our external state. I feel hungry, I'm going to go get some food. We regenerate, we reproduce, we replicate. So to understand replication, we need to know something critical about life, and that is that life builds from the bottom up. Most of the objects that we have in our houses are off the shelf. We don't build our televisions, our cell phones, our stoves from the bottom up. We build them off the shelf complete. But what life does is build it from the bottom up. When we eat, we break our food down into its component parts, proteins broken into amino acids, carbohydrates into simple sugars, and then we build everything from scratch from those components, proteins, blood cells, muscles, organs, etc. Now, there are a couple of exceptions which are just curious, but I think in the long run unimportant. For example, there's some nudibranchs, I'll show a picture in a moment, that reuse, for example, the stinging cells of things like Portuguese men of war to help protect themselves by ingesting them. So here's a Portuguese man of war. There's one of its stinging cells, nematocysts, and they're ingested and incorporated into the body of the nudibranch. So they're borrowing an organized structure and using it directly, but that's strange in the animal world. Okay, so how does life copy itself? Building from the bottom up means the need for instructions for making proteins and all the other molecules. All living organisms achieve this using DNA, and there's a machinery for taking the instructions and converting them into protein and all the other molecules. Okay, so now let's talk about the key steps in the origin of life, at least life as we currently know it. Stars provide the elements, the Big Bang, etc. I'm not going to deal with that. There's making the building blocks. There's concentrating the building blocks. There's making insides and outsides. There's establishing the replication machinery, and there's the harnessing of energy. And those are the essential elements. And so what I want to do now is walk through each of these things, the extent to which we understand it or don't understand it. Making the building blocks. Prior to 1952 or so, the study of the origin of life was pretty much theoretical in people's head. And then a graduate student at San Diego, Stanley Miller, performed a very simple and elegant experiment. What he did is he made guesses as to what the Earth's initial atmosphere was like from gases that come out of volcanoes. So he made these gases, he collected them in a glass container over here, and then he zapped them with lightning. And then he collected the products of that zapping. And what he found is that he could form a whole range of fairly complex molecules quite quickly. And some of those, the ones in reds here, are actually amino acids that are essential to your body and all other organisms today. So it turns out that making the precursors of life turn out to be remarkably easy if there's energy flow. Now, some molecules are harder than others. So we now believe that the composition of the Earth's early atmosphere was not that used by Urey and Miller, so fine. Lightning is also pretty violent and sporadic, so we probably need some sort of energy source. And you still have to concentrate the building blocks, and the atmosphere is probably not a good place to do that. And it does seem harder to build the components for DNA and RNA than, say, for proteins or lipids. But a fellow called Sutherland in the UK has produced some very elegant experiments generating on the chemistry bench, RNA molecules, for example, which is crucial for life. So important progress is being made in that area. The key point is when energy flows through a system, interacts with it, thermodynamics predicts that there will be increased order, that is, that there'll be organization and complexity. Driven systems explore the improbable. That's what thermodynamics actually says when energy is flowing. And it turns out, even in interstellar space, there's enough energy to interact with the sparse molecules out there to form complex molecules. There's 200 types of molecules been identified. The largest has about 60 carbon atoms involved. This diagram molecule diagrammed here, anthracene, is one of the few that actually hasn't been properly confirmed yet. So energy flow, even way out there, produces organization. Okay, we've got to concentrate the building blocks. So we need either restricted environments or a way of actively concentrating the building blocks. Oops, wrong direction. So understanding how the building blocks were concentrated takes us to a central question. Where exactly did life originate, do you think? And what I want to do now is actually ask you a question. So what I do in my undergraduate classes is that we hand out bits of colored card to everybody, and we ask questions like that, and we get them to hold up their cards so we get a sense of where people are thinking are. So if you wouldn't mind typing in the chat your favorite color or colors as to where you think life might have originated. In small evaporating lakes, in geothermal hot springs like Yellowstone, in the open ocean, in deep ocean hot springs, in outer space or in other planets, or somewhere else. As I'm watching the answers come in, lots of greens, some blues, red, a yellow, orange, purple, excellent. Okay, fantastic. Yellow in the open ocean. Good on you, Richard. Okay, I'm not going to give an answer yet, but I will. So let's just explore just for a moment the hypothesis that life actually originated somewhere other than on Earth. What's the motivation for the idea? Part of it has been that maybe it's so hard to explain the origin of life on Earth because it didn't happen here. Another possibility is it might be true. For example, if there was always rich oxygen ever since we began, then I would have to go for something like origin of life elsewhere, because I don't think life can originate where there's oxygen pervading everywhere. Assuming life did originate elsewhere, and we don't think it did, not to say that it doesn't exist outside, how did it get here? And so it turns out that we now have about 126 meteorites that are derived from Mars. A meteorite struck the surface of Mars, blew Mars rocks into orbit, then it orbited around and eventually fell to Earth. And the chemical composition clearly indicates they're Martian in origin and not from Earth itself. And some of them were up there for a long time. So there is potentially a vehicle for getting things from the surface of Mars to the surface of Earth. And that's really fascinating. I don't think it's especially important for the origin of life, but it's there. Okay, so concentrating the building blocks. For some, clay is an attractive model for concentrating the building blocks. They can serve as templates for the formation of larger structures. And here's just a cartoon showing that. Here's sort of an isolated clay crystal with various molecules being washed around by the liquid. And the organized structure of the clay provides a template for the organic molecules to accumulate and may help catalyze their polymerization into more complicated molecules. We're not sure that clays were important, but we do think that mineral surfaces were probably very important in forming early molecules at the early stages of the origin of life, and we'll get to that in a moment. Okay, making insides and outsides, and how to get the key molecules inside them. So it turns out that making protocells, making insides and outsides, is actually pretty easy. All you need are linear molecules that have one end that's oily, fatty, and the other end that's water soluble, indicated by the circle here. Chuck them all in water, and the oily ends want to get away from the water as much as possible, and they self-organize into these spheres. In fact, here there's two layers of these molecules that looks an awful lot like the way your cell membranes are built, at least crudely speaking. The ease with which these can be made suggests that the formation of cells, cellularization, was an early step. But Clef and I have had a problem with this is that, remember, life is dynamic. It's a verb. There has to be a constant flow of energy and material through it, in and out. And it's not at all obvious how you got the right molecules in and how you get the energy flow to operate. We'll get back to that shortly as well. Okay, establishing the replication machinery. And here's a case where where progress has been spectacular. So there's a really nasty chicken and the egg problem associated with establishing replication. Cells need protein to read and replicate DNA, but DNA is needed to make the proteins. So today's organisms can't make protein without the DNA, but you can't make the DNA without the proteins. So how on earth did replication get started? And for a lot of people interested in the origin of life, this was just a complete stumbling block. And people who wanted to study it, like Cliff Brunk, couldn't because there was nothing to do tomorrow because we couldn't get around this problem. This all changed with the discovery of ribozymes. RNAs are known to be information storage molecules, but it turns out it was discovered they also can do chemistry. They can function. And it's now hypothesized that the first organisms were organized around RNAs, both for information storage and for doing chemistry. And then later, information storage was taken over by DNA and most of the metabolism by proteins. So here's a little cartoon. This is a simple RNA strand. It wouldn't be straight, it'd be wobbly in the water, and another RNA that's interacting with it. And when the base pairs match up just so, it is able to do something quite magical, and that is to break a bond, break the RNA in two. This very simple experiment earned Sid Altman a Nobel Prize, and along with Tom Cech, also a Nobel Prize winner, led to the idea of ribozymes in the RNA world. So this bit is understood quite well, the origin of replication. An analogy would perhaps be the modern orchestra. The music is made by the instruments. They're the analogs of proteins, the hardware that does the work. What do they play? The music itself is encoded in sheet music. That's analogous to the DNA. But prior to either instruments or sheet music, music was simply sung, and that's analogous to the RNA. Imagine a musical world where no one could sing. Well, this is the modern world of biology. There are no longer any RNA-based organisms today. All we have is the equivalent of the instruments, the proteins, our protein bodies, and our DNA that codes the instructions for what how those proteins are to be made. The last piece of evidence for the RNA world, which I think has proved decisive for most people, is that even though most of our chemistry is performed by protein and our information storage is DNA, RNAs are intimately involved in the steps between DNA and its conversion into protein. This next diagram is a little complicated, but it's fairly simple in principle. I start with DNA in red, and I finish up with protein in red, and the little black dots are the individual amino acids that are joined together to make the protein. In order to get from DNA to protein, the DNA is converted into messenger RNA, which is threaded through the ribosome, which is largely made of RNA, stabilized by proteins, and the amino acids are brought in by tRNAs. The amino acids are joined together in the protein actually by one of the RNAs in the ribosome. So it turns out while most of the chemistry in your body is done by protein, the chemistry that actually makes the protein in all organisms is performed by an RNA. We have an ancient relic of the very first organisms at the heart of our very own biology still. And it turns out that sort of argument, these baked in anachronisms, is going to be important towards the end of my story. So now we come to the harnessing of energy. There's a very strange two-step process that we use to generate our ATP. ATP is the energy currency inside all cells. It's kind of like the money in the cells. Most of what happens is mediated by the energy of ATP. And the two steps are pretty strange. The first step's fairly straightforward. We burn sugar to provide energy to pump protons across our mitochondrial inner membrane. And then those protons in a high concentration fall back through this blue blobby thing, which actually generates the ATP. And as it synthesizes, it's called ATP synthase. So it's like we have a coal power plant burning sugar, pumping the protons like into a dam, which then flow back to run a hydroelectric plant, ATP synthase, to make the ATP. Why this crazy two-step system? Why not just burn the sugar directly and use the energy directly, which we can also do, for example, when we produce lactic acid in our muscles. Oh, final point, which is important. The low concentration of protons in the middle means a high pH. The high concentration of protons on the outside is low pH. I know it's counterintuitive. It's the way it worked. It's going to be crucial at the end of the lecture. Okay, so where do we think life originated? So to answer that question, I'm going to ask you a very strange question that might seem completely irrelevant. And that is, how long do you think the longest continuous mountain chain is? So here are your choices. If you, again, you can type your answers in the chat. The Earth's circumference is about 25,000 miles. So your choices are 400, 1,600, 4,300, 40,000 miles. And so the question you might also answer is, what's the name of the mountain chain that's the longest? I'm seeing lots of different colors. The only color I'm not seeing is red. I'm seeing lots of yellows and oranges. So it turns out the answer is 40,000 miles. So what are the mountain chains? There's the Sierra Nevada, the Himalaya, the Andes, which is the longest terrestrial range, and then the mid-ocean ridges. And many people don't know about them because, of course, they're buried under the ocean. So here's a picture of them here. I'm going to start up here in the Arctic, and it runs down right through the middle of Iceland and down through the middle of the Atlantic. That's why it's called mid-ocean ridge. And what I'd like you to note here, there's a groove, there's a valley right in the middle of it, all the way around. And then across between Antarctica and Africa, and then and on it goes, 40,000 miles of it. So what creates it? Why is it important? So it turns out the mid-ocean ridges are geothermally active. They're the largest geothermal source of activity on the planet. So if we cut away the planet here, what we see is that the heat from the core drives circulation of the mantle, which comes to the surface, and molten rock pours out onto the surface, actually creating the seafloor. This is the valley here. As the seafloor moves, it eventually drops down again, creating earthquakes and volcanoes and tsunamis like in Japan. So seafloor is being created, and that's the valley where seafloor is created in the middle of the mid-ocean ridge. And so in the next slide, I'm going to show you what that looks like, what happens at the geothermal activity there. So we're going to go to an ocean ridge off the coast of Oregon and Washington. There's the valley sitting in there. And then submersibles then discovered these famous black smokers, producing highly toxic, extraordinarily hot waters coming directly from the molten rock, heating the seawater underneath this black smoker. And what was remarkable with these discoveries is it was replete with life. The first cruises had no biologists. They didn't expect to find life down there. And what's striking is it's completely dark. And so it led to the realization that life doesn't need light. We use light to produce chemical energy that then drives ATP synthase. I can just use chemical energy directly without light, which is what these guys are doing via bacteria. Too nasty for the origin of life, too acidic, too toxic, too hot. So not a good candidate. But the way it operates, which will be important in a moment, is there are fissures and cracks in the seafloor where seawater flows in, is heated by the hot rock underneath, which then comes to the surface again, producing the black smoke. And here then is one of those black smokers taken with one of these submersibles. So what someone did around 2000 was looked for geothermal activity, Goldilocks hydrothermal events, off the mid-ocean ridge, where the water is going to be cooler and less violent. And what they discovered was the following. And so here we have towers, chimneys being built. You can see the hot water flowing through, but it's clear now, not black smoke. It's much cooler. 40, 60, 80 degrees Celsius, and nutrient rich, which is why there's so many organisms there. And so it looks that these structures are the structures that led to the origin of life. And so this diagram here, it doesn't show the fissures in the seafloor, but again, seawater, which will have been slightly acidic 4 billion years ago, interacts with hot rock, and the rock now is mantle rock, which then creates these chimneys or vents. So far, so good. That's nice. And then the staggering discovery was made, and that is that these chimneys, these towers, are just perforated with lots and lots of micropores, microcatacombs. And what we think is that this is where life began, inside these microchambers. And in a big tower, there could be billions of microchambers. They're so small and the towers are sufficiently big. And there are hundreds or thousands or tens of thousands of these spires scattered across the mid-ocean ridges, particularly 4 billion years ago. So the pioneers in this was Mike Russell, who at University of Glasgow discovered fossil equivalents of this in Ireland. And he recognized based on the minerals that the temperature was right, the pH wasn't acidic, it was alkaline. And he proposed then in the late 80s that this might be the cradle of life, a decade before these things were discovered in the real world. And then Russell and Bill Martin in Dusseldorf have been the ones who've been largely pushing the idea. And then Nick Lane spending more attention on the energetics involved. And so these are the three big names. And what I'm going to try to do now in my last few minutes is summarize the core of the ideas and why we find it so compelling in terms of an overarching framework for understanding the origin of life. So we think the origin of life was probably in the micropores within these alkaline hydrothermal vents. There's about 1,000 times more CO2 then compared to now dissolved in the seawater. There is hydrogen gas coming out with the interaction between the seawater and the hot rock. The only shared metabolism between all bacterial and archaeal groups uses CO2 interacting with hydrogen gas. So it actually mimics, looks like the first biochemistry. There are minerals, iron sulfide, that look like they were necessary for polymerizing and catalyzing the first formation of complex molecules. So we think the mineral surfaces played a crucial role in establishing the chemistry of the first molecules. And you can see here in this cartoon, they only have half a dozen microchambers rather than 500 million microchambers. But what they're arguing then is that you get increasingly complex molecules, in some respects using the height of the microchamber as a proxy for time. So there's a steady flow of energy, hydrogen gas, and raw materials flowing into the chambers all the time. It's the fabric of the structures of what actually makes the microchambers. The micropore walls provide initial delineation of inside and outside. I don't have to provide it from anywhere else. And then I bet you no doubt it accreted quite quickly lipids and things, and you had crude protomembranes forming on these solid rock walls or these porous rock walls. The micropores probably enabled high concentrations of biomolecules. It turns out there's a process called thermoparesis where as the water flows through, the faster water takes away the smaller molecules and the slower streams off to the side will, of course, a concentration of larger molecules. And you can get 1,000-fold increases in concentration really simply over just a few micropore distances away. The other thing, of course, is they're tiny, tiny, sort of cell-sized, roughly, which means you don't need much material to have high concentrations. You don't have to fill up a huge pond, you don't have to fill up an ocean, you don't have to fill up the atmosphere to get biologically important concentrations. And then the other thing that makes it especially attractive is there was a proton gradient, a pH gradient across the chimneys, the spires. The pH of the water was slightly acidic at about 6 or 7. The pH of the water coming in was somewhere between 9 and 11. So that means that there was roughly a 10,000 times higher concentration of protons outside than in, which would have naturally then flowed in to the microchamber spires. And so that to us makes sense, to these guys who invented the theory, makes sense of the way we make ATP today. We have the chemical energy all along, which we still use in our biology, but there's a second energy source that's available if you can just tap it, a natural pH gradient. And so it looks like ATP synthase was able to capitalize on that pH gradient, giving you a decisive increase of energy and therefore reproductive advantage compared to things that didn't so. And so this two-step process makes sense given the alkaline waters and the acidic waters. So finally, two really interesting surprises. Archaea and bacteria have the same genetic code down to the ridiculous details of the 64 codons. They have exactly the same details of the protein synthesis machinery, etc. So they clearly had a same ancestry in the same chimney, but they've got different compositions of their cell membranes and also their cell walls. They've also got other differences I'll mention in a moment. So that suggests that they cellularized independently, that LUCA, the last universal common ancestor of life, was not cellular, simply using the microchamber walls as its enclosing entity. And so if I draw a diagram uniting the bacteria and archaea, it looks like the last universal common ancestor was sitting down here before the advent of organized cell membranes in pink here and green here on this diagram. DNA is also interesting. Archaea and bacteria use different processes and different proteins to replicate their DNA, a little bit like different operating systems, Mac OS or DOS or whatever it is. So that suggests that they developed DNA independently, and thus that LUCA was not DNA-based either. So it suggests then that two of the defining features of life today, cells and DNA, was unlikely present in the common ancestor of all of us that are currently alive today, which I think is really particularly interesting. And this microchamber hypothesis makes sense of those differences that we've seen between bacteria and archaea. Okay, I'm doing well on time. I just want to come to sort of a very interesting sort of almost philosophical consequence of this theory, if it's true, is when we look at typical trees of life, there's the animals there and the fungi and the plants, and there's the archaea and there's the bacteria. Each lineage is depicted as a single line. And the way evolution works in a Darwinian classic way is, I have an innovation which I pass on to my offspring. They might have an innovation which they pass on to their offspring, et cetera, et cetera. So innovation is called vertical descent. You have to pass your innovations on from one generation to the next. And as a consequence, it's very, very slow. But when you think about the way human society operates, the way we learn, there's lots of what's called horizontal transfer. I learn from my parents, my brothers and sisters, my uncles and aunts. I learn from my schoolmates and my teachers. I learn from my university professors and my classmates. I learn enormous amounts of information that doesn't rely on me to learn it individually from one generation to the next. So it's called sort of horizontal transfer, and it leads to enormously rapid evolution and innovation. And so what we think happened at the early beginnings of life is there was similar vast exchange of innovation. They're all in the same enclosed space. Innovations will have percolated through the microchamber complexes. And maybe one of the reasons why life succeeded early is because of its human-like capacity, or I'm not sure, to share information, to share ideas in a sense, new molecules, new ways of processing, etc. And even when they escape from the microchambers, bacteria are quite good at exchanging genetic material with each other. And it really isn't until we get more complex organisms that we finish up with this really glacial rate of evolution. And then, of course, we say with human evolution, now we're back to incredibly rapid innovation with the way we exchange ideas and things. Okay, if the white non-smoker idea is correct, and it or bits of it might not be, it implies that LUCA was not cellular, but was cradled inside the micropores. The cell membranes and DNA were among the last features of life to evolve, though no doubt the micropores were coated with some sort of membrane-like structure early. That life evolved in the pitch black, and that life arose as a consequence of tectonics, the interaction between warm rock and seawater. It really is truly Mother Earth. The attractiveness of the ideas is that the cradle of life are long-lived and geographically extensive. The mid-ocean ridges over geologic time can be viewed as a massively parallel process which had a consequence of producing life. And it wouldn't surprise us that an RNA world type life originated hundreds, thousands, millions of times. We don't have to rely on a freak environment to explain the origin of life, a warm little pond. What if it dried out before life could work out how to leave it? The chambers have built-in pH gradient, a proton gradient, which makes sense of life's bizarre way of generating ATP. And it solves the problem of how to concentrate the building blocks and how to get them inside the first cells, because there was energy and material flow through the microchambers, which were, in fact, in a sense, the first cells. So we now feel we actually have a framework, which is where my excitement is coming from, for understanding the origin of life, even if we don't yet have all the details. And I'm sure there's an awful lot we have to learn yet. And so with that, thank you for your attention. And I think I have left time for questions.
You have. Thank you so much. That was wonderful. I'm going to now, just like I said, if you have a question, you can go ahead and raise your hands using the reaction button. And also we have a series of questions we'd like to ask you, Charles, that came in from the audience. But I want to encourage people to continue asking questions, because there did seem to be quite a few questions that were asked that were eventually answered. So Charles, you had a really great story, and you had said before that if we interrupted you, it likely would come up later. So I'm glad that you mentioned that, because a lot of these questions were answered. But I'm going to go ahead and hand it over. Maybe Thomas Klein can unmute himself and ask a question if you'd like.
Yeah, it's not quite the origin, but I've always been intrigued with photosynthesis and how long it took for oxygen-producing photosynthesis to evolve. And it occurred to me recently that when the planet was much hotter and full of water, is it possible there was really extensive water cloud cover all over the Earth that would have made photosynthesis not all that active? It may have been going at some rate, but it wouldn't have been going well enough to actually convert the iron from Fe+3 to +2 and precipitate, which took about 500 million years. But I'm just wondering what's known about the atmosphere of the early planet, in particular water vapor, and whether there could have been really dense water vapor clouds.
So I think the answer is yes, there well could have. And even today in climate modeling, we're not very good at clouds. Thinking about them and understanding their extent of them. You know, low levels of light are low levels of light, and they're everywhere. And so just being able to harness light just a little bit would be an enormous advantage. And so I think as long as there was any ambient light and a source of chemical energy like iron sulfide at the beginning, photosynthesis would have been a huge gain. To me, the real innovation is the second type of photosynthesis, where you actually use water now as the electron donor, not iron sulfide, because then you can do it almost anywhere.
Yeah.
First photosynthesis is stuck still to hydrothermal type systems, but then you can, whenever there's water and sunlight, you can live, and that's almost anywhere on the surface, which is fantastic.
Thank you. That was great. Jim Stauffer, do you want to go ahead and ask your question?
Yes, this is a great talk. Are there active or planned experiments to reproduce the kind of environment with the microchambers and the chemicals and the pressure and whatever?
That's a really good question. And so when I teach this, one of my concepts is sort of the what are you going to do tomorrow test. And if you can't work out what you're going to do tomorrow, it means that the science is really immature. And so one of the beauties of this is it leads directly to immediate experiments. And so, for example, Nick Lane and others have started to build microchambers and explore the pressure, the temperature, the chemical gradients to start to see what they can grow. So this is now active underwork that is underway. And to me, that's what makes it really exciting now, is most of the components now of the story of the origin of life are now amenable to experimental procedure and testing, where until the microchamber hypothesis came along, the RNA world was going great guns, and it is, and extremely difficult and important problems. But the rest of it was kind of sitting there waiting for some sort of framework to start designing those sort of experiments.
Thank you. And Jeff Greenspan would like to ask a question. So go for it, Jeff.
Well, I have several in chat that maybe, because I don't have a raise hands option.
Why don't you
the
go ahead
question-
and pick one of your
Yeah,
favorite
that's what I'm
question
gonna
for
do.
now?
That's what I'm going to do. You did answer several of my questions, which are how would membranes have developed, and would the LUCA have been in an RNA world or in DNA world? So I'm going to ask you the question, when the genetic code started to develop and amino acid formation started to go to a tRNA amino acid translation system, do you think they developed in tandem, and that was an active system even before all the amino acids got to be formed, so that the transfer RNA system and the amino acid system developed in tandem, and you could see primitive peptides forming before the whole genetic code formed? Over.
Yeah, so good question. So amino acids are formed very early, and I suspect more of them than we currently use. And amino acids form protein or proteinoids pretty easily, which have a lot of functions. So we think that even before RNA, proteins were already being produced catalyzing reactions, but it was hit and miss because there was no way of reproducing it. And so then you work out how to make an RNA world, and then you work out how to bind an amino acid to a specific RNA and then join them together. So it's a fairly complicated process, but we think, as a paleontologist, one of the things that's always struck me is almost all major innovations involve multiple steps of pre-adaptation. Feathers are key for bird flight, but they originated presumably for insulation and or display well before there was flight. And this happens over and over again. So almost everything that was important for the evolution of life already was being made previously, and then it gets co-opted or refined in some sort of way. And so I think it's really important to realize that in the RNA world, while a lot of the chemistry is being done by RNA, I think the cell walls are being lined by lipids, protomembranes. They're also being lined by proteinoids, so amino acids and proteins. And so all the different components are there, and it's sort of systematizing it. That's the key innovation, which replication gives you, the RNA world. So I think I'll stop there before I get out of my depth and too complicated.
Thank you very much. Thank you very much. Do we actually have evidence of prebiotic catalytic activity in hydrothermal vents? Over. Last question.
I think people have demonstrated, for example, iron sulfide minerals help catalyze reactions. And then it turns out ferredoxins, there are a whole bunch of proteins today that have exactly iron sulfide or iron nickel sulfide structures, just like the ones in the pores that are part of the essential enzymes that we use today. So again, it looks like we have those baked in anachronisms that date back way back to the very beginning before even RNA.
Thank you.
That leads to another great question that we had, and you, I think you might have touched on it a little bit, but has the hydrothermal vent origin hypothesis been duplicated in the lab?
In a sense, no.
Mm-hmm.
Because if all of it had, then I think we all would have heard about it, because that would be truly sensational. So
Yes.
So people are trying different bits of it.
Mm-hmm.
Like,
okay.
can I get the acetyl-CoA pathway to develop abiotically in the lab, which is one of the first carbon fixation pathways. So life is sufficiently complicated that each of the steps, and no one, and this is crucial, has been able to produce, I think, RNA in that setting yet. And we think what's going on is that I think, one thinks of a cell being water and the microchambers being filled with water.
Mm-hmm.
Cliff and I tend to think of them more as gels. They're packed stupid full of RNAs and proteins. There's water squeezed in. So the chemistry inside that's very complicated and not very simple aquatic chemistry that one thinks of initially when one thinks of prebiotic experiments.
Can I ask a follow-up to that?
Sure, go for it, Michael.
It sounds kind of like Nick Lane is planning these experiments out.
He's been publishing on them. Yeah. So the names I gave you, Russell, Martin, Lane. If you go to Google Scholar, look them up. There are lots of reviews and experimental papers. It's fantastic stuff. Those three names are the key big names.
Thanks.
All right, let's head even farther back not farther back in time, but I mean farther back looking at the bigger picture. There were a few questions on viruses and how viruses fit into the tree of life? Are they on a branch? How do they fit in that way? So several people asked this, so maybe you can talk a little bit about that.
I did see that and I thought, yes, of course. Viruses are tricky.
I think viruses are a hot topic right now.
Yeah, so we don't really understand. So there's two sort of extreme possibilities, is that they represent an early form of life or a form of life that originated who knows when, that by being able to be parasitic on the cell's machinery have given away most of their primitive structures. And all you have is little relictual sequences that are able to get themselves into your genome so that you can reproduce them and they don't have to bother doing it. And they can use your metabolism because they don't have to bother doing it. The other possibility is that they're fairly late and that they're sort of parasites that worked out how to do exactly the same thing. And the problem is there's so little information in them that we really just can't trace their history through the analysis of their genes or their sequences. And so we really don't know. The sense is they had to be fairly early because parasitism occurs almost immediately in biology. As soon as there's a resource, there's something to exploit it. But how early with respect to RNA world and things, there just doesn't seem to be any good evidence to answer that question. It's a really good one.
Great. Those of you who asked, that's a good question. So then,
so Ezra, my good friend, asked, are they monophyletic? Do they have one origin? And the answer is we don't know. There isn't enough information to tell. Might be multiple origins, hundreds of origins, one origin, don't know. Sorry, Dion.
No. I think that was like our next question on our list. And you saw it and you answered it. That was great. So the next question that had to do with viruses, and you did. So the question straight up, are they alive? It sounds like yes and no.
This is really tricky, right?
Yeah.
And so there's this issue, I think Dawkins actually coined the term extended phenotype. We pride ourselves on being individuals and independent, which is completely stupid because we're dependent upon energy flow and material flow. You know, out of oxygen, I'm dead quite quickly. If I don't eat, I'm dead quite quickly. We depend in so many ways on the environment. So viruses depend crucially on their environment because they can't replicate themselves. And so I think we were discussing it with the grad students on Monday when I sort of went through a rehearsal of this. And one of them said, and I think I tend to agree with him, which is, I think the debate as to whether they're alive or not itself is fairly sterile, but having the debate is enormously revealing in terms of what we understand life actually is. And I think the connectiveness and the fact of life being a verb, not a noun, helps understand that. So I'm sorry I can't do much better, but they are very tricky.
No, I think that's great. And it sounds like maybe also this person alluded to maybe in some situations you might consider, some definitions you might consider them alive and other definitions you might not.
Yes.
Great. And then there was also some comments and questions about habitable planets, and just doesn't this imply that there is a likelihood of extraterrestrial life then?
Oh yeah, so I took the slides out at the end because I thought it'd be too distracting. So what about 1 in 5 stars now look like they have an Earth-like planet around it? It looks like if you're rocky, then you almost certainly have tectonics. It looks like Mars had it for about 1 billion years. It's got giant volcanoes, it's got rifts. And so if you have plate tectonics, you almost certainly have geothermal belts. And so you have geochemical energy being produced. You have water, which is probably crucial. You need to have carbon, but that's one of the most common elements in the universe after hydrogen and helium. And so I think life is probably pretty ubiquitous, simple life. Cliff and I have these arguments as to whether RNA is the only molecule that can self-replicate. It may be one of the very few, so we suspect that life may actually have something like RNA-like. You've got to have something that's a cast in a mold of itself. And that's one of the few molecules that does that. Proteins can't do that, lipids can't do that, etc. But I think it suggests that relatively simple life. Now, really important, life today, once you develop oxygenic photosynthesis, transforms atmospheres and ocean. You can measure it from afar. And once you invade land, you can, well, even the ocean, you can see it by change of color. But when life is sitting in those microchambers, it's basically invisible from space. It hasn't escaped yet, its volume is small, its geochemical cycling is small. So I wouldn't surprise me if life occurs on almost all Earth-like planets that have water around circling stars, but I can't think of a way of detecting it with the sort of technologies that NASA's developing it.
Why the obsession for liquid water? I can understand the liquid part because you need a medium in which molecules can float around and interact with each other. But what's so special about water? Why couldn't it be another element, another compound?
Yes, what's special about water? So one, it's very common. Two, it's liquid at a fairly high temperature, which affects the rates of reaction, which is important. And then it forms hydrogen bonds. It looks like it's really, really crucial. But I know as an undergraduate chemistry minor in PChem, we did an exercise, for example, liquid ammonia has a lot of the same qualities that water has. It forms hydrogen bonds. Right. It's way, way colder. So yes, it needn't necessarily be water. It's so easy to fall into the trap of life as we know it becomes the only way that life can be. So I appreciate being checked on that.
Thank you.
And similarly, and I liked this question, and it goes back to the water piece. So seeds in the pyramid were doing nothing for 3,000 years and then germinated when watered. Were they alive or dead when inactive? This is to counter example your description of life with water, right?
Yes, but also I was arguing that life, a fundamental quality of it is energy flow and material flow. Mm-hmm. And obviously anything that insists, tardigrades, lotus seeds, they're 1,000 years old. While they're in the insisting stage, they're static. They're held stationary.
Yeah.
And so that's where the verb comes in again. So that the continuity of history. Yes. And most of our definitions are not embedded in the historical context and history of the object you're talking about. So we remove ourselves from history with the way we do abstract thought, and then it produces these paradoxes. So to me, those paradoxes tell us that we're not thinking correctly about it. So that they're really important to think about. So I love that.
Great. Hey, Zachary McGuire, if you want to take yourself off mute, you can go ahead and ask your question.
Thanks. So if plate tectonics is a necessary component for life to develop with the geochemical energy being thrown through the ridges, do you think that places like Europa or Io that have plate tectonics, but not necessarily water or carbon-based biochemistries can form? Like something like silicon or boron?
Okay, so I'll go two places first. The first, of course, is it looks like there are persistent geothermal vents, say on Enceladus and Saturn. And that's tectonics driven by the gravitational squeezing of the moon. So it doesn't have to be Earth-like in that sense. So thank you for correcting me on that. The problem with silica, which I thought about a lot, carbon's special because it can form 4 bonds, but it also forms bonds with itself easily. Silica forms most of the minerals, but it often is silica, oxygen, silica, oxygen. And one of the things about silica that's really annoying is it's not gaseous and it doesn't dissolve well, where carbon dioxide does both well. So carbon is easily transported and moved. Silica is not easily transported or moved, and it doesn't seem to form nearly the same number of compounds. Doesn't mean that you can't build life around silica, but it looks like that's probably why it didn't. It just doesn't have those special qualities of dissolvability, transportability, and the range of molecules that it can form. I've got a very couple of quick questions I can answer very rapidly, which is how does red algae sit in the evolution of things? There's plants, animals, fungi, then the red algae produce the kelp, then they're golden algae, brown algae. So are now up to six multicellular groups of eukaryotes, and so they go on. So they're an independent evolution as distinct from plants, animals, and fungi as we are from each other. And then how did life replicate before RNA? It didn't. The molecules were generated de novo over and over again as the engine of the energy flow and the hydrogen and the CO2 coming in, but it didn't replicate until RNA. So the RNA for us represents the beginning of life, the beginning of replication. And once you have replication, then you have Darwinian evolution. because you have small modifications, and if that causes an increase in the rate at which you replicate, your molecules will be increasingly represented, and therefore the composition of the molecules in the microchamber changes to send with modification. So for us, replication is crucial for evolution, which is crucial for refinement, and you don't do that until you get RNA.
What is the ramification of the creation in lab of a completely new amino acid that has been done?
I don't think it's so important. You know, there are lots of different types of molecules. They're used in different ways. Life is enormously innovative. There are relatively few molecules that are truly special. Yes, we have molecules then that we become dependent upon, but if they'd been replaced at the origin by a different one, then that would be the one that's dependent upon. You know, so, you know, I hope I'm special to my daughter, but if I never existed and she never existed, there will still be lots of very special parents and children out there. So I think the specialness of individual molecules is not so important.
Here's a great bit of an abstract question we got earlier about, can we define life or any living system that fights against entropy instead of properties like complexity or reproduction, and that are also shared by non-living systems? How do you have thoughts on entropy and
yeah, so that I think entropy is a red herring.
Nice.
And I didn't express the words that way, but the slide I put in saying when there's energy flow, when the system is driven, you expect increased order. I didn't say material cycles and complexity. Well, that is anti-entropy locally.
Yeah.
Now you're creating entropy elsewhere to create the energy flow, so the net entropy is increasing. And then what I didn't say, which I think is truly special, and I haven't seen in the literature, is energy flows in, and it's like a billiard ball, makes a molecule A, interacts with B, makes C, D, E, F, running down as it using up energy. But life early on invented energy storage molecules, ATP, and then you can inject energy when it's needed. And so it's like having a little puff of air to keep a feather up. And you think of a Rube Goldberg machine. We got things going point, 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 point. Little bits of energy get injected all the time, rubber bands, little bits of gravity. And so I think then you think of biochemistry, things are constantly being injected with a little bit of ATP to keep the thing going and cycling and going round and round and producing all the pathways. So that is sort of negentropic, driven by the energy flow coming in from the burning of the sugar and the generating of the ATP, which is net system entropic. So I think for life itself, entropy is pretty unimportant. We've broken the constraint of it, particularly with energy storage molecules.
Got it. Those are some strong thoughts on entropy, so I'm glad I asked. Thomas Klein, did you want to come back and ask a question? I think you re-raised your hand. Is that correct?
Yeah. Let's see. Yeah, I just had a suggestion. I was intrigued when you're defining life because it seemed like you were trying to define all the things that life has to do because of this definition. And when I taught genetics, I always started out with the definition of life. And it struck me as being incredibly simple. It's basically reproduction by the directed transformation of the environment. And that distinguishes it from crystals and everything else. And particularly transformation or reproduction by transformation of the environment, it's subject to Darwinian evolution because it has to be a way of doing that that can evolve. Obviously, RNA does that, but the question is, would there be any other way of doing it? But if you simply say you have to transform the environment to reproduce yourself, that seems to me, obviously the virus are alive. They transform the bacterial environment into more of themselves than our environment. But it's a very simple definition that I could suggest.
I like that a lot. It's somewhat similar to NASA's definition, which is something to the effect of a chemical system capable of Darwinian evolution.
Well, that really strips it down.
but it includes the fact of replication and modification
and change.
But really, what I like about your definition, which I've not seen, is you've almost by definition included the inside and the outside in the definition.
Yeah.
And that's fabulous, rather than trying to define it independent of the environment. I love that.
And the other thing I would add is that if you throw in Darwinian evolution, then the ubiquitous feature of life is it expands into new niches. It basically is opportunistic. And
Yeah,
that's why
explores the improbable.
That's why I see us as the only way that intelligent life can get out of the solar system because basically and get to all those wonderful new niches out there for us.
Yeah, I love that a lot. The other thing now, interesting, what I'll say is that, and the definitions have their uses, and that I love that.
Yeah.
But I saw a criticism of the NASA one, which is depending on what you want the definition for. If you want to teach genetics and cell biology, sounds like it works brilliantly. If you want to use it to help discover diagnostics to discover life on another planet,
Yeah.
maybe it's not so helpful.
Well,
so some of the astrobiologists are not so happy with the NASA one because it doesn't help them identify diagnostics, for example, to test whether or not there's life on Mars or on exoplanets.
no. But I would argue you start with the most simple, the obvious. And since it happened once, that's the most obvious. So look for that. And besides the fact, I'm somebody who believes life didn't start here. I think if you look at the evidence, it's just staring you in the face. It couldn't have evolved here, even with the smokers. It takes so long to do everything that seems trivial compared to the origin. And so if it came from somewhere else, then we go to Mars and we look for our kind of life, because that means the contamination, we just as easily have contaminated Mars as us. But anyway, that's my editorial.
I don't want to start a debate, but the problem is you've got to start it somewhere.
Absolutely. I like your smokers, but by the way, can I ask, have people gone into the smokers and looked for the molecules that are in those little microcells?
People have generated them in the lab, and I don't know the extent to which people have put probes directly into them and measured.
Yeah.
But there's certainly, there's methane, there's CO2, there's, I think there's acetate. A lot of the small 2,3 carbon molecules are measured directly.
Mm-hmm.
Now, someone says here, how long ago did the geothermal processes start on Earth? And I think pretty much as soon as the crust formed. And so the Earth's about 4.6 billion. And the oldest evidence of any sort of crustal material is about 4.5 now, 4.4. The oldest zircons, I don't remember the date. So we think that crustal processes may be, 4.4, 4.5 billion. But it looks like there are occasionally large meteorites that zoomed in that may have vaporized the surface of the Earth. So I wouldn't surprise me if life got started and wiped out multiple times until the bombardment of this large material dropped low enough that the Earth became continuously stable.
Can you address the recent hypothesis that also came out of universities in California that life might have originated in freshwater, like hot springs environments?
Yeah, so I think this is tied very much to Sutherland's work, where what they find is if they have cycles of desiccation and hydration using ultraviolet energy, then they can produce a whole bunch of molecules, including RNA and amino acids, pretty simply and straightforwardly. And the ideal place for that would be on the edge of a pond or geothermal pond. And you've got ions, which seem important for protein folding, magnesium, potassium, etc. You have low sodium, which is probably a good thing since most organisms don't like sodium. And so I think it's Sutherland's experiments where he's actually been able to generate RNA easily, kind of as easily as Miller did, or sort of easily, that's led to rejuvenation of the freshwater pond hypothesis. And I don't want to dismiss that, despite the appeal I have for the microchambers. Now, a lot of the cell is relatively dry. And so one of maybe the early role of lipids and proteinoids early on was to make dry spaces. And it may be that you can do desiccation, drying in the microchambers, even though they're in water, because you can reduce the activity of water, particularly if you've got tiny, tiny spaces. But no one's demonstrated that. So to me, if you could demonstrate RNA using the sort of chemistry that we see hypothesized for the edge of freshwater ponds in a marine setting in these microchambers, then that would be huge in terms of pushing the microchambers theory ahead.
Thank you.
Hey, Charles, did you see the question about expecting to see the processes taking place today along the ridges, or are the theories rely on very different conditions existing only in earlier times? That has to do with kind of the conversation we're having now.
Yeah, I did not see that, but that goes back to my early slide, which is, first of all, oxygen is in relatively high abundance. So it's just going to burn, oxidize things, rust them really quick. And the second one is that all these organisms there taking advantage of any precursor molecules that are, you form amino acids, they go, hey, great, free food. So I think with the oxygen and with existing organisms, I'm a pathetic, slow, cranky organism that can barely live. I'm just going to get wiped out. I mean, rates of reaction with protein are like thousands, tens of thousands of times faster than RNA. And then if you have oxygenic photosynthesis, you've got 10 times the energy. And so these first beasts were barely alive by our standards and would have just been eaten alive by anything that lives today. And so that's why I think the world is so fundamentally different from it was then that no, I don't expect life in those environments anymore. And I think life, when it developed these things, just wiped them out.
Yeah. So Robert Penny's question about free oxygen levels being low enough, no. Or no, they are not low enough.
I don't think.
Yeah. Yeah. These are all.
I don't think. No, so I don't know. So that's a good question. Can you produce anoxygenic conditions inside the vents? And I actually don't know the answer to that. But we also have anaerobic organisms, archaebacteria and bacteria, that are very happy in anaerobic environments. So even without oxygen, there are things that can snavel up resources. Lots of things. You know, the stinky smell you see in San Francisco Bay at low tide, you know, hydrogen sulfide producing, you know.
I'm sure some creatures love that smell, so.
Yeah, so one of the interesting questions we have here, could all these theories are correct and took place simultaneously?
Yep.
Early on, I thought clays was daft because there's nothing clay-like, but the mineral surfaces look like they're important. So clays are sort of alive, except maybe they're not clays. And then in the RNA world, maybe you produce RNA on the edge of these things and it's next to the marine setting. And so they, I don't know. So in some pure sense, no, they can't all be right. But maybe there's a way of linking them. And I was looking through the literature, it turns out there are microchambers, tiny ones, formed of silica in Lake Taupo, which is a geothermal lake in New Zealand. So maybe it's possible to have freshwater microchamber type setting. I still want to have a pH gradient to explain that, but maybe there's other ways of generating it. So I really don't, you know, I've got good friends, my supervisor from Chicago, David Rout, now passed on, or Walter Alvarez, sort of, you know, bolide impact thesis at Berkeley here. And I remember talking to these guys when their theory started to get going with the meteorite that wiped out the dinosaur hypothesis. And both of them said, as I was developing the theory, I started to get really scared because I started to believe it. And once you start to believe in your ideas, you often lose your critical edge, which is why it's so crucial that they're competing ideas, that there's a review process, that they're independent reviewers. You know, power corrupts intellectually as well as all the other ways. So these questions in terms of poking the multiple things, I think are absolutely crucial. So I appreciate them. I also apologize, we're not going to get to all the questions, but that's, you know, sorry.
Yeah, no, I think that's a great way to end. And I feel like we did actually hit a lot of the questions and all the questions through the chat we've been sorting through. And that's actually a nice poetic way to end the lecture. Our audience has been incredible, don't you think? Lots of really very brilliant questions that even have, you know, you scratching your head like, this is a great question. That's a great way to think about things. So I'm so happy to have this conversation with all of you. I'm so happy. Let's thank Charles once again for being here. It has been such a delight. I'm not sure if I mentioned this, but the reason we wanted Charles here today at this time during this month, do you want to talk about it, Charles?
Oh, just the 100th year?
Yeah.
Yeah, so it turns out that on March the 14th, the Museum of Paleontology on the Berkeley campus, UCMP, which I'm the current director of, celebrated 100 years of existence. And so part of this actually was to celebrate 100 years of paleontology at Berkeley. Not that this is quite paleontology, but it is the broad sweep of the history of life. And I just thought, since we have a moment, the slide behind me is paleontologically enormously important. There are hundreds of thousands of fossils in those rocks. and there's the Cambrian explosion of animals, but the world has relatively few species in it. They've got small body size, there's not many individuals. And then in the Ordovician, the marine realm starts to fill up, and you can walk up the slope behind me and watch the species richness, the species diversity, the abundance, and the body size just increase as you walk up this hill slope over 5 or 10 million years of geologic time. And then there's a 30 million year basalt on top, which is irrelevant. So yes, so thank you. Yes, so we've just celebrated 100 years.
Yes. So, and you guys have events and websites and things dedicated as well. Is that correct? To the 100th year articles. Yeah. And I think I've been seeing some things go through. So if you want to check out the website to learn more. So thank you again. Thank you all of you for joining. See you next month. So thank you. Bye, everyone.
Thank you very much.